discuss about the medical genetics. The genetics that is applied in the medical field, that genetics is known as the medical genetics. Actually, it is the application of the genetics. Then what is genetics? Genetics is nothing but is the branch of biology that concerned with the study of genes or genetic variation. Actually, from name it is clear, it is the study of the gene. What? It is the study of the nothing but is the study of the gene. So, first is the Mendel, who gave the term genetics. Who? Mendel. Mendel is applied the genetics in the pea plant. You know that what is the Mendelian genetics? Mendelian genetics is the first genetics. Actually, Mendel he is a farmer. Mendel is a farmer. He see that there are a different color of the pea. So what happened in the pea plant, that they appear different in different pea plant. So he think and apply the term genetics. At that time he don't know gene is the main reason, that that's why there is a difference between the flower color. There is difference between the flower color or height. So he gave that term as a factor. What? In Mendelian time, gene is known as what term? In that time, this gene is known as a term by factor. What he do? He select the different color of different flower of the pea plant. Like he select red and white, or he cross red pea plant to red flower pea plant, and he observed that after selecting this both red color, then he got three different varieties phenotype of the plant, or two different phenotype of the plant, that is red and white. In the ratio, what did that ratio? 3:1 that time. So he think that there is a in plant or in organism that control this all things. That time he only think this term and gave the term this substance or thing which control this all color, height of the plant or organism. He gave that term as a factor. What? He gave that term factor. That factor nowadays, this Mendelian factor is known as gene. And this gene, the study of this gene or their genetic variation means their variation comes under this gene, and study about that variation like deletion of the some sequence in the gene, or addition or mutation, recombination. There are so many factors come under the gene. So that study about that genetic variation in the gene, that this is controlled under the study of genetics. So what is the medical genetics? Medical genetics is nothing but it is applied genetics, application of the genetics. The genetics that are used in the medical field, like as a therapeutic in making of the drugs, that is known as the medical genetics. Okay. So medical genetics are the applied genetics. Nothing but it is the applied genetics. So today we study in this medical genetics. In medical genetics we cover all the unit 1. Okay. So in unit 1, first topic is recombinant DNA. What do you mean by recombinant DNA? From name it is clear that it is the combination. What? Recombination. Recombinant means combination of the different sequence of the DNA. We combine the two different sequence. Suppose this is one DNA, this is 2 DNA. We have to make a combination of these two DNA. Then what we do? We cut something here, we cut something here, and then we seal it, right? Combine it, seal it. For this cutting and sealing, what we need? Enzyme. What we need? Enzyme. And for this transferring, what we need? Vehicle. What? For transfer the things from one place to another, what we need? Vehicle, right? Here also, and this vehicle is known as here what? Vector. What? As a vector. We call this vehicle as a vector. We need vector, enzyme, vector, right? And for sealing, that enzyme is known as ligase. What? Ligase. So, for recombinant DNA, for the combination of this, then we get a DNA that is the combination of this is 1, this is 2. This both DNA sequence contained in a single strand. This type of DNA is known as recombinant DNA. What? Recombinant DNA. So, recombinant DNA is a technology that uses enzyme. What uses? That uses enzyme to cut and paste together. What? First, cut. Second, paste. Paste together. Sequence of interest. Sequence of interest. Suppose we select two organisms. Okay. Suppose we select 2 bacteria. Right. Bacteria. This is first bacteria. This is second bacteria. This is a thermophilus bacteria. Okay. And this bacteria are a cold resistant to cold. Okay. This is thermophilus. We have to select this thermophilus bacteria heat resistant genes, and we have to select here cold resistant gene. Okay. Now what happened? We want a organism that have both resistant, cold and heat both resistant. So what happened? What we do? We isolate the heat resistant gene here. We isolate the cold resistant gene here, right? And then we cut some gene here and select your interest of the gene, means cold resistant genes and heat resistant gene. Then we seal it. This is cold resistant gene. This is heat resistant gene. And then what happened? This is called recombinant. What? Recombination. Okay. So this technology is used for our purpose. What we want to get, right? So the recombined DNA sequence can be placed into a vehicle. What? Vehicle called vector. Vector. Already I told you. Here we need an organism that transfer. We isolate DNA sequence already from an organism, and we need another organism who transfer the gene from one place to another place. And that organism which help in the transferring, that is known as vector. That to ferry the DNA into a suitable host where it can be copied or expressed. What? Where they can be copied or where they can express, right? So this is the diagram of recombinant DNA technology. How restriction enzyme cut, how ligase seal, how vector are helping transferring the DNA sequence. So here we understand clearly. This is the donor DNA. Donor DNA means the DNA sequence from the organism who donate their DNA sequence. That organism is known as donor DNA. Okay. that organism is known as donor organism, and the DNA from that donor organism is known as donor DNA. This is the donor DNA, and here it is a vector. What? Vector, which help in the transferring of the donor DNA into the host cell. Okay. Remember one thing, that the enzyme which cut the DNA, which cut the DNA, donor DNA, that restriction enzyme also, same restriction enzyme are used to cut the vector DNA. Remember? Means restriction enzyme are used, right? Restriction enzyme are used for cutting. What is the purpose of restriction enzyme? They help in cutting the DNA sequence, right? So, restriction enzyme are used for cutting the DNA sequence. There are 3 different type of restriction enzyme. There are types of restriction enzyme, 1, 2 and 3, right? So, here what we need, the restriction enzyme. Suppose EcoR1, we use EcoR1. This is a restriction enzyme. Okay. This restriction enzyme are used. This restriction enzyme, if cut this restriction donor DNA, then this EcoR1 can cut the vector DNA, not other. Okay. Remember this. Single restriction enzyme are used for both donor DNA and vector DNA. Okay. So, here this is a donor DNA. We use EcoR1 to cut we cut this DNA. Okay. Then we get a sticky end. This is a sticky end. When restriction enzyme cut the DNA, there are 2 type of end will be produced. First one is sticky end, and second one is blunt end. Blunt end. Sticky end means there is a overhanging strand are present. This is a sticky end, means another come and stick here. And next another one is blunt end. Blunt end means this is a DNA. Here they cut, and they produce this. Means there is no flanking regions are present. No flanking regions are present. Blunt end are present here. Okay. So, what happened? See, EcoR1 are used. EcoR1 cut this donor DNA, and here sticky end are produced. What? Here sticky end are produced. This is a sticky end. Okay. Same EcoR1 cut this vector. This is a circular DNA. Or plasmid. We call it plasmid. What is plasmid? Plasmid is nothing but is extra chromosomal DNA. Okay. So this EcoR1 cut this vector, and here also a sticky end are produced. See, this is a sticky end. Okay. Now, next enzyme are used, that is DNA ligase. What is the function of DNA ligase? The main function of the DNA ligase are what? They help in ligating or joining. They help in joining the DNA. Joining the DNA, right? The main function of the DNA ligase is the joining of the DNA or seal overhang. So, they combine with this overhang region and form a complementary region with each other, and they join with each other. DNA introduced into a, when they join, see, this is this blue region is a what? Donor DNA. This blue region is the donor DNA, and this red region is the vector DNA. here there is no role of vector sequence. Only the sequence we have to isolate, that is of donor DNA. Vector DNA only act as a vehicle. They help in transferring the donor DNA into a host cell. Now this DNA introduced into the bacteria. Generally we use bacterial cell, because bacterial cell are they replicate fast. They replicate fast. What? They replicate fast, like the binary fission. They replicate by binary fission, right? Means 2 to the power 1. So we used bacterial cell. We introduced this recombinant DNA or this donor DNA into the bacterial cell. This is the donor DNA. Here this is a donor DNA. Here this is a donor DNA. Now this recombinant DNA molecule replicate and cell will be divide. So now what happened? This is the combination of recombinant DNA. Now you can isolate. When we need this recombinant, this sequence, we isolate this sequence from this bacteria, right? What? We isolate this sequence from the bacteria. And we also make a clone. What? Make a clone. And there is a library. There is a gene library where this clone are stored there. When we need the sequence or some other gene of our interest, we go and isolate that clone. Okay? Understand? Recombinant DNA technology. So, here what are the tools used in the recombinant DNA technology? There are mainly four type of tools that are used in recombinant DNA technology. That is DNA polymerase, second is restriction endonucleases, third is DNA ligase, fourth is cloning vector. What is the function of DNA polymerase? DNA polymerase is nothing but they catalyze the synthesis of DNA molecule from the nucleotide triphosphate. From polymerase, what do you mean by polymerase? Polymerase is a enzyme. What? Always remember, if their age is come in last, means they are enzyme. Here see, restriction endonuclease, ligase, this all are a enzyme. If A's are come in the last, that means this is a enzyme. So polymerase is nothing but is a polymer. What they do? They polymerize the DNA molecule, right? This is a small fragment of DNA molecule. If DNA polymerase will be present, what happen? They polymerize or make a long DNA molecule. Okay? Catalyze the synthesis of the DNA molecule. What is the function of the DNA polymerase? Catalyze the synthesis of DNA molecule. Okay? By what they used for when they catalyzed? Simply, DNA are made up of nucleotides, so they use the nucleotide. Actually, there are three different type of DNA polymerase. DNA polymerase 1, 2, 3, okay, 4. Sorry, there are 4. This is a different type of DNA polymerase. Mainly the polymerase which polymerize the DNA molecule is the DNA polymerase 3. What? DNA polymerase 3. Remember one thing also. DNA polymerase always polymerize the DNA molecule from the 5' to 3' end. Always in the 5' to 3' end. And they repair from the 3' to 5 prime end. Repairing will take place from the 3 prime to 5 prime end. Repairing. And 5 prime to 3 prime, polymerization. Polymerization will take place. Okay. So, DNA polymerase is the enzyme that catalyzes the synthesis of DNA molecule from the 5 prime to 3 prime end. Okay. DNA polymerase at the nucleotide at the 3 prime end of the 3 prime end. Okay. What they add nucleotide at the 3 prime end, because at 5 prime end phosphate group are present and 3 prime end OH group are present, right? So DNA polymerase need primer. What? They need primer for the polymerization. So another enzyme are present, that is called primase. Primase is the enzyme which synthesizes the primer at the site of the DNA polymerization. When primer will add there, then DNA polymerase go and bind with that DNA primer and they polymerize the DNA molecule. So this is the function of DNA polymerase. And if there something wrong nucleotide will be substituted at during the polymerization, then there are another DNA polymerase 1, 2, 4 that help in the repairing of that DNA, that nucleotide. Okay. Like DNA polymerase 1, they repair the DNA or correct the nucleotide or remove the primer from the 3 prime to 5 prime end, right? So for all things there are different enzymes are present for their different properties. Next is restriction endonucleases. What? Next is second one is restriction endonucleases. What is the function of restriction endonucleases? The main function of restriction endonucleases is the cut the DNA. Nuclease. Remember, nuclease. Nuclease has the property to cut. What? They cut the DNA molecule. Okay. So what happened? Endo means they cut the DNA molecule from the midpoint. There are two type of restriction endo. First is there are two type of restriction enzyme. Actually, they are a restriction enzyme. See, this is a restriction enzyme. Restriction enzyme are of two types. First, restriction endonucleases, and next restriction exonucleases. What? Endonucleases and exonucleases. So endonucleases cut the DNA from the mid, and exonucleases cut the DNA from the end. Okay. So main property of the restriction nucleases is the what? They cut the DNA. When recombination of the DNA will be takes place, there is a need of the cutting of the DNA so that we seal the or join the both vector and interest of DNA, right? So we need to cut the DNA molecule. So then who cut that DNA molecule? That restriction enzyme. Restriction enzyme cut that DNA molecule. And same restriction enzyme are used for both interest of DNA and vector DNA, right? Same restriction enzymes are used. So restriction endonucleases are of three types. 1, 2 and 3. There are 3 types of restriction endonucleases. We go in next slide and study in detail. Okay. about the restriction endonucleases. Next is DNA ligase. What they do? DNA ligase is the enzyme which help in joining there are only one function of DNA ligase, that what? Joining the DNA molecule. What is the function of DNA ligase? Joining the DNA molecule. One and only on one function. Next is cloning vector. What is the function of vector? Already know. They act as vehicle. They help in transferring the DNA molecule or recombinant DNA from one place to another. Okay? Understand? This slide is clear? So, come. Restriction endonucleases. What is the restriction endonucleases? It is a bacteria enzyme that recognize and cleave foreign DNA. What happened? That recognize and cleave the foreign, suppose this is a foreign DNA, they recognize the sequence, because each and every restriction endonucleases have the specific sequence where they go and cut. Okay? Each restriction endonucleases have a specific sequence. They not cut anywhere. Means this is the DNA and the restriction enzyme come and cut anywhere. No, not happen like this. Restriction enzyme only cut the DNA from the specific DNA sequence. Okay? So, restriction enzyme recognize that their specific sequence, if that sequence are present in DNA, suppose this A, T, T, G, C, right? Restriction enzyme cut at this GC region. Okay? And here this. Okay? So, they come recognize this sequence and cut at this point. Not anywhere else in that DNA sequence. Okay? So, recognize and cleave foreign DNA. This is the main function of the restriction enzyme. There are three types of restriction enzyme. First enzyme, they cleave DNA at random sites more than 1 kb from recognition site required ATP. Means type 1 restriction enzyme, they need energy. What? They need energy for cut the foreign or DNA sequence, foreign DNA sequence. Okay, foreign DNA. They need energy as a form of ATP. They need energy as a form of ATP for the cut the DNA sequence. Another important things is they cut at random site. What? They cut at random site more than 1 kb from recognition. Suppose this is a recognition site, then they cut at the 1 kb, between 1 kb randomly, anywhere. But they recognize their sequence, this recognition site. They recognize their sequence, and from that sequence they cut randomly 1 kb, from 1 kb. Okay. Next is type 2. Type 2 cleave DNA within recognition site, do not require ATP, most require divalent cation. Means type 2 recognition site, they not require the energy. They not require energy. Type 2 recognition site cleave DNA within recognition site, do not require ATP, most require divalent cation like Mg, magnesium ion. Magnesium ion is a divalent ion, they require magnesium ion, not ATP. Not ATP. And they cut the DNA within the recognition site. Means this is a recognition, ATTGC is a recognition site, then they cut within this ATTGC. Nothing away from this and nothing, no, they not cut away from this recognition site. They cut within the recognition sites. So, type 2 restriction endonucleases cut the DNA or cleave the DNA within recognition site. Remember this. Type 3 cleave DNA at random site approximately 25 base pair from the recognition site, and they require ATP. Same as restriction endonuclease first, means they require ATP and they cut randomly away from the 25 base pair from the recognition site. Type first, what happened in first? Type first cut the DNA from away 1 kb from the recognition site, and type 3 away from 25 base pair from the recognition site. The restriction enzyme that are mostly used in recombinant DNA technology is the type 2. What? That is type 2. If there any question will be asked, which restriction enzyme are mostly used in recombinant DNA technology, then you said that type 2. And why? Another question they ask, why this type 2 restriction enzymes are used? Why these restriction enzymes are used? Then you say, why this type 2 restriction enzyme are used? Then you say that because type 2 restriction enzyme cut within the recognition sites. That's why this type 2 restriction enzyme are mostly used due to their cutting reason. All this first and third cut away from their recognition site, but type 2 cut at the restriction within the recognition sites. See here, this is the diagram of restriction enzyme. This is the DNA fragment, double helix DNA fragment, and this is, suppose this is eco R1, right? Eco R1 and eco R1 cut the DNA sequence, and they cut at like a sticky end. They produce a sticky end. And eco R1 is a type of type 2, means they cut the DNA sequence within the recognition site. This is the recognition site, then they cut within the recognition site. And same restriction enzyme are used in both DNA fragment. Now they produced a flanking region, and this flanking region joined with each other and form a recombinant DNA technology, right? Form a recombinant DNA technology. So, next one is DNA polymerase. Already we discussed detail in DNA polymerase. So, here we see that how DNA polymerase polymerize the DNA molecule. This is the DNA molecule, right? This is the DNA molecule. This is template strand. We call it as this as template strand, and this is newly synthesized strand. Okay. So remember that DNA polymer is always polymerized from the 5' to 3' direction. Always remember this. So new strand will go in this template, in the direction of template strand, and nucleotide will be added here, and they polymerize the new strand. This is the function of. And one thing also, if primer is not present, then DNA polymerase will not work. They work only when primer will present. First primer, this is the yellow region is the primer. The primer will add, then only DNA polymerase come and bind, and they polymerize the newly strand DNA. Okay. This is the strand they polymerize. Next is gene cloning. What is the gene cloning? Gene cloning is you know the technique PCR. In this COVID-19, all person will know what is the PCR, right? PCR test, this is a PCR test or random test, PCR test. Like what happened there? In PCR, their clone will be formed, right? Same here, gene cloning. Okay, scientists allow to make a large amount of DNA from small fragment. What happened in gene cloning? In gene cloning, we produced scientist, lab technician, or they all use gene cloning to form a large amount of DNA from small fragment. Okay. By gene cloning, we used, clone means what? We used same thing, like same thing. That is known as clone, right? So in gene cloning, we clone the gene. What? We clone the gene in gene cloning. Yeah, this is the diagram of how to make the clone, how to make a gene cloning, how to produce the cloning of the gene. Actually, cloning will also produced with the help of recombinant DNA technology. Gene cloning is also application of the DNA technology. This is the plasmid and this is the human cell. Okay, we want to isolate the growth factor, gene of growth factor. We want to isolate the gene of the growth factor, right? This is the growth factor gene. Now we used what? Restriction endonucleases. We used EcoR1. EcoR1 cut this and we isolate this plasmid from the bacteria, because bacteria only contain the plasmid, that is extra chromosomal DNA. EcoR1 and restriction enzyme cut this plasmid vector. This act as a vector and produce flanking region after cutting by the restriction endonucleases, that is EcoR1. They produce flanking region both. Now DNA ligase are used to recombine this both plasmid and gene of human growth hormone. After combining this gene for human growth hormone, we want to clone this gene. What? We want to clone or increase the number of this gene. How we can increase? We insert this gene into a bacterial cell. What? This is a bacterial cell. We insert the gene into this bacterial cell. What happened here? What happened here? The bacteria cell will divide. When they divide, then the plasmid will divide. Plasmid will also divide, and they increase their copy number. So this increase with the formula 2 to the power n. 1 to 2, 2 to 4, 4 to 8, like this. Okay. Now we make a clone. What we produce? Clone. Okay. This is the method of gene cloning. Next, the types of cloning. What are the types of cloning? There are 3 types of cloning. Okay. First is reproductive cloning, second is therapeutic cloning, and another one is DNA or gene cloning. What happened in reproductive cloning? In reproductive cloning, we make a clone of whole animal or human. Like we propagate transgenic or genetically modified organism, like dolly, sheep, right? Like dolly, sheep, you know, everyone is know. This is that is the reproductive cloning. Next one is therapeutic cloning. Therapeutic cloning means we used or harvest the stem cells embryo, right, to cure the disease that are used in many pharmaceutical companies. And next one is DNA or gene cloning. In DNA or gene cloning, what happened? They make the copy of gene or DNA, and that are also used in pharmaceutical or industrial in industry. Okay, for making the GM organism. Okay, These are the three types of the cloning, that is reproductive cloning, therapeutic cloning and DNA or gene cloning. Next one is what is the application of the gene cloning. First application, human insulin production. This is the most famous and the earliest application of the recombinant DNA technology, or we say that of the cloning. Not we say that cloning, but this is the main application of the recombinant DNA technology. What happened here? We isolate the gene. We isolate the human insulin producing gene. Where human insulin will be produced? Pancreas, right? Pancreas is the organ where insulin is produced. Okay. Then we isolate the insulin producing gene from the pancreas. Okay. Isolate the gene from the pancreas, and we isolate the plasmid from the bacteria. Cut that gene and vector with the restriction endonucleases and recombine that DNA with the plasmid vector, right? And form a recombinant DNA. Then we add this recombinant into a bacterial cell. Now we want a large scale of this insulin production, right? This is not only produced by the bacterial cell. We want large production. So what? We use fermentation technology. We use bioprocess technology, right? We use fermentation tank. We transfer this recombinant bacterial cells into a fermentation tank, where recombinant bacteria multiplying and producing human insulin in a fermentation tank. After large amount of human insulin was produced, this is the insulin, right? This is the insulin. We harvest this insulin and used in a pharmaceutical companies as a drug. Okay. This is the application of the recombinant DNA technology. There is another application that is not, actually that application is not used in a medical field, but is also a most common application, that is insect pest control. In this we use Bt gene. What? We used Bt gene, Bacillus thuringiensis gene, that produce the desired lethal protein is joined to a marker gene for antibiotic resistant. What? They produce antibiotic resistant. Bt gene plus marker gene inserted into the plant. What? Here we isolate Bt gene. Here this is a marker. We join this, recombine this. What? Recombine this, and this are transferred into a plant. What? Where? Plant. Plant cells are grown in the presence of antibiotic. Cells that carry the Bt gene plus antibiotic resistance gene survive and are grown into a plant. Okay. Here we add that antibiotic. Okay. What? This plate contain antibiotic. If there is a gene that resistant to antibiotic, they grow in this plate, and the gene which are not resistant to antibiotic, that not grow, and we isolate that resistant gene with Bt content and we transfer into a plant cell. Okay. Then that plant will be resistant to that pest. Okay. Bacillus thuringiensis. So these are the two application. Okay. So here unit 1 is complete. So thank you.